So, I have done two of these streams today. The first one I did a bit of media revision. Second one I did some RE revision, and honestly it kind of blew my mind the amount of stuff I'd forgotten. I think when you assume something will be easy, you just completely reduce your perspective on it to a point where you begin to forget even the most obvious of details. And I think that's what happened with RE. I forgot a lot of common stuff that I should have known. So anyways, let's get right into it. We're going to be doing science, which means mainly chemistry and biology. I first want to start off by doing, I guess, you know what, let's go back all the way to like year 7 here, right? Let's go all the way back to year 7, and let's just do a simple look at the cell, because on one of the papers that me and Ashton looked through, there was literally a question on drawing a cell, yeah? So literally just like drawing a circular shape with a nuclei, ribosomes, things like that, mitochondria, obviously the cytoplasm, which I guess could just be a colourless blob, and then obviously the cell membrane, which controls what goes in and out of a cell. Now, an important thing to remember about the mitochondria is that the mitochondria releases energy. It does not create or produce energy. It releases energy via respiration. Okay? So remember, respiration, the equation for that, if I'm not mistaken, is glucose plus oxygen, and we get water, CO2, and energy, or ATP. And that is how the most energy is created. Now, a good question to start you guys off with today relates to exactly what I've just talked about. Let's assume that we have a test tube like this, okay? And I'll kind of make this a little like darker at the bottom here. Okay, so we have a test tube. Okay, let's assume there's some like pondweed in it, and then let's also assume that there is a snail in it. Okay, there's a snail. So let's assume that our graph in terms of carbon dioxide concentration, so this is time, and this is CO2 concentration. To begin with, it's going to be basically like this, okay? But then let's assume that the snail dies. This happens. Why is that? Well, number one, why is it flat here when a snail and algae are inside this test tube full of water? Why is the carbon dioxide level completely straight? Well, think about it logically. The snail releases CO2 due to respiration. That CO2 is then absorbed by the algae and is converted into oxygen and glucose via photosynthesis. The oxygen is then used by the snail for respiration, etc., etc. It's a cycle. No carbon dioxide is lost or gained. It remains a constant until the snail dies. And when the snail dies, think about it, it's going to be on the floor. And what's going to happen is decomposers, such as bacteria, are going to break away at the snail. They're going to eat it. And when this happens, CO2 is released because it is being consumed by the decomposers. Parts of the snail are being consumed, which is then used as energy. Okay, so when it's eating, that is then creating energy, which is used for respiration, which then releases CO2 as a result. So in the exam where this question was a thing, a lot of people wrote the fact that due to decomposition, it broke down and released CO2. But why would it break down? It doesn't just break down naturally, especially in water. The reason it would is because there's decomposers there. Why would CO2 get released? Because those decomposers are respiring, which releases CO2. Hope that helps you guys there. Talking about respiration, bioenergetics. Okay, let's move on to some actual other stuff. We have the structure of a cell here. Let's just draw a leaf. You guys already know what we're going to talk about here if you came on the last stream. The structure of a leaf. So you have many various features in a leaf. You have the row of palisade cells. And for those unaware, the palisade cells are right at the top, and they contain a lot of chloroplasts. And why would that be? Why would there be a shit ton of chloroplasts right at the top of a leaf? Or an epidermis, I don't know how you pronounce it. The skin, the top layer of the skin of the plant. Why would there be a bunch of cells containing chloroplasts? Well, obviously, in order to absorb as much light as possible. Because think about it, if the sun's up here, it's releasing light, then if they're all the way down here and there's all these other organelles and cells, not much light is going to get through. So they're right at the top so that the most amount of light is absorbed. They also have a vacuole I believe vacuoles are for water exchange, things like diffusion. And then they also have a cell wall to help with structure, because they do exist right at the top. So that's the palisade cell, very important cell. Let's talk about the spongy mesophyll, or the spongy cells they're called, but they form together to create the spongy mesophyll. So these things have lots of gaps. They are sparsely packed or loosely packed, and for that reason, they are perfectly adapted for gas exchange. They also have a thin layer of water on them, which means that the gas can diffuse in and it is absorbed by the water and taken to other parts of the plant. So they are very effective for gas exchange, okay? And they are spongy. It's such a goofy word, spongy mesophyll. I love it. Next are the guard cells and the stomata cells. They're a bit like little pores in the plant, and the way they function is that they allow gases to exit or enter the leaf. So for example, it allows CO2 to enter, because CO2 is used for photosynthesis, and the byproduct of that, oxygen, can be released. Happy? Guard cells just control the stomata. Okay, finally, we have the root hair cells, but they are kind of like, I'm gonna draw it kind of squarey, so then you can kind of have the vacuole. So they have a large vacuole because they are adapted best for water exchange, I believe. Things like diffusion or osmosis primarily. Also active transport. They have cells with a lot of mitochondria in, so active transport can occur a lot. And that is good because obviously that means you have a strong concentration gradient, and it means that there is a constant supply of water being taken to other parts of the cell. The root hair cell absorbs water, that water is taken to other parts of the cell. Also mineral ions too. So it's a bit like the villi, except, for a plant. One thing I wanted to talk about were the meristem cells. I don't know how these things look. I imagine them to look a bit like this. I don't know really. I don't know how they look. But these things are found in the roots and the shoots. Now, what are merry stem cells? Well, it's kind of in the name, merry stem. So when I first read this word, I was like, okay, it must be something to do with growth, because stems, that's kind of for growth. I honestly forgot stem cells. They are the things that differentiate into other cells in order to allow for, like a child, a fetus, for example, there's the embryonic stem cells, and then there's the adult stem cells. These are the plant stem cells. And I did not know that. I genuinely did not know that, but that could be many marks. And I don't think I've ever learnt that in school, honestly. I don't know, you guys will have to tell me if you remember meristem cells, but it could be in the exam something about plant differentiation. But they are found in the roots and shoots, because obviously those are the areas where the most growth is going to happen. The roots, because that allows water and things. Speaking of which, you have the xylem and the phloem. These two, I used to never be able to tell the difference between, but now I do. So let's go through. Okay, number one, the differences between them, we'll talk about. We'll do like 3 for each. So the differences between the xylem and the phloem is that the xylem is for water and mineral ions. It takes it from the roots to the leaves, because the leaves need it for photosynthesis. They need water. And also, the xylem is made of dead cells, and it is supported by, I think it's called lignin? Lignin? I don't know if that rings a bell. Some kind of lignin, some kind of word like that. It's strengthened by that, I believe. I can go check my revision book if you guys don't know. And then in terms of the phloem, oh, and I forgot to say, the process is called transpiration. And then we have translocation, which is where, obviously, the glucose / sugar is taken from the leaves to other parts of the cell. Well, the plant, sorry. Because remember, glucose isn't going to be much use in the leaves. It needs to go everywhere, because glucose is used for growth. It is used for aerobic respiration. It is used for a lot of parts of a plant. Actually, here's a question, if anyone is watching. I can't really tell if anyone's commented anything yet. Nothing's popped up for me. But I don't know really. But here's a good question that you guys could answer. What are 3 uses of aerobic respiration? Like, what's the purpose of it? And by that, I'm not saying, oh, it makes glucose, it makes, I mean, like, what are the uses of a product of aerobic respiration? And then finally, the phloem is made of alive cells. And I'm not too sure what else you could say. I guess the fact that, they allow sap to leave. They have like pores to allow sap to escape. To get back onto that question that I just asked, because I don't know, again, if anyone's actually watching the stream or not. Some uses of the byproducts. So, for example, glucose. What's the purpose of that? Well, obviously, aerobic respiration. And what's the, purpose of aerobic respiration? Well, that releases energy, which is used for active transport, movements, you could say. And I'm trying to think of another feature. I guess maybe cell growth could be one. Let me go check my revision guide to make sure I haven't missed anything from that section. But I think that's everything, guys. If you're happy, I'm happy. And you guys are going to like my next talk, because it's going to be something pretty fun. Let me see. We've talked about the stomata, mesophyll, palisades, epidermal, which is the skin. Transpiration is called the transpiration stream. If you put transpiration in your exam, you won't get it. Cell sap is a liquid made up of the substances being transported. Okay, so phloem, I was right. They transport food substances. And the most obvious one that I forgot is the fact that the xylem can only go one direction, the phloem can go both directions. Okay. So, I feel pretty confident with that. Guys, I prepare food at college. I think that was our acronym from last time. Oh no, it's I prepare meals at college. Mitosis, okay? We have interphase, prophase, metaphase, I think it's anaphase, and is it cytokinesis? You guys will have to let me know if any of that is right. I'm assuming it is. So interphase is where the chromosomes in the nuclei replicate. Prophase is where the nuclear membrane breaks down. Metaphase is when they line up in the middle and the spindle fibers attach. Anaphase is where they're pulled apart. And cytokinesis is where a new membrane is put around each set of chromosomes to create new nuclei, and the cell is pinched to create two identical cells. How dare you leave me on red? I sent you a message. So there you go. If any of you guys did not know that, seriously, it honestly could be worth 12, 15 marks in an exam. You could get asked why mitosis occurs, so you could talk about cell repair, allowing the immune system to, continue, cell growth. And you also should probably talk about the fact that they are checked for errors as well. If you get a blended question where it talks about like cancer, you could definitely refer to the fact that occasionally errors may occur, such as, the incorrect amount of chromosomes being created, I'm guessing. Let me know when you're ready. You said like PlayStation 5 on your PC or something? I mean, dude, I've been live for 20 minutes. I'll be live for like another 40 or something. I'm doing a massive review of everything, so maybe have a tad revise yourself if you haven't already. And then I should be able to do it. I'm just making sure that I'm ready, because remember guys, the biology exam is the second exam that you do, maybe the first if you don't pick RE. So the first exam you're doing, and okay, seriously guys, pay attention here, right? I know for a fact that a lot of people suck ass at certain parts of biology. I know for a fact that not too many people know the food tests. And this has been proven time and time again. Mr. Hooper asked our class a while ago about food tests, and not too many people got it. I also checked the examiner's report, and this is a question that 1 in 5 students got no marks on, which means a lot of people don't do well at food tests. Enzymes links directly to food tests, digestive enzymes, and I know for a fact that you guys could not name every single enzyme, its byproduct, and how it attaches. Also, I guess the lock and key method, that's an important one. And I think as well, a lot of people don't know mitosis. I know for a fact last mock exam, Ashton, I didn't understand it. Ashton said he remembered the acronym, but not what any of the steps to. I hope that by watching these videos, these streams, that you guys now know what mitosis is. I hope now that you guys know what the food tests are, and I hope you at least know one or two of the enzymes and the lock and key method. If you guys don't, then I would strongly advise you to go on the Google document. If you don't have access to it, send me an email requesting access or whatever. It is very useful, and at least for me, it's allowed me to remember so many things about these bloody questions and stuff. It's mad. Well, that's Mr. Hooper. No, but like, I'm saying he asked the class and no one knew. But this is a thing we were taught, like, in year 8, year 9. He went through it with us in a revision session. And he taught it relatively well. And then the fact that no one a week later was able to know it. I mean, yeah, Mr. Hooper isn't the best teacher, but even still, that either shows that A, everyone forgot it, or B, just people didn't put their hands up. But even still, that means that they must be somewhat unconfident with it. So yeah, I don't know, should I go through the food tests? Oh, have you got like Chat GPT to generate you a meme or something? Dude, I kid you not, though. I'll ask this. I'll ask this. I'll ask this completely, like, if you don't want to say, it's fine, because I know a lot of people who don't. But do you do much revision yourself? And I'm asking that because I'm intrigued to see what other people are doing for revision. Like, seriously, guys, do you guys do much revision? Because I think Kieran does a tad bit. I think Ashton does some. Ellis probably does like an hour a day or something. But I don't know too many other people who... No, 0. Oh my Jesus Christ. Guys, on a real note, if I know obviously it is getting pretty concerning how quickly we're getting towards these exams. Like, bloody 6 days away now, before our first exam. But not 6 days, sorry, 8 days technically until biology exam. But on a real note, guys, if you need any help with biology or chemistry, check the Google document. I will keep repeating this until I die, but the Google document is the most useful thing I've ever made. It genuinely contains so much information, links to the past streams, everything like that. It is good. If you don't have access to it, send me an email on my school account saying, invite me or whatever. No. Shit, for real? That's wickety whack. It is, bro, it is. The fact that we have bloody... I mean, for me, I have 2 days, 2 school days. All of you guys have 4? Yeah, Tuesday, Wednesday, Thursday, Friday. But I have this bastard media exam that I already did, but I have to redo. So I'm missing 2 days of revision, and I want to die as a result, because I don't like media anymore. Which is ironic, because media's like the only thing that I do, but in terms of like videos and stuff. But no, screw the media exam. It can burn. Yeah, I mean, people like I think Bobby, Katie, like Lewis maybe. Don't think James, Danny maybe. I think they all don't have to resit it because they like got a distinction or higher. But because I was 1 mark off hitting my target grade, I have to resit. And it makes me cry because it is horrendous. I could be doing actual revision and I have to sit in an exam hall with invigilators all day, here, writing about a bloody magazine that I'm never going to make. Person sitting in front of a computer with a confused and frustrated expression while JMC Arts YouTube channel plays on the screen. But okay, thank you for that. Anyways, what do you want me to go through now? Any suggestions, guys? We've just gone through mitosis. We've just gone through cell structure, plant structure. We could go through the heart of a digestive system. Yeah, I got a mirror in IT, and I don't want to redo it, but I was the last person. Ah, damn. That's horrible. I mean, she kind of forced me to reset it too, honestly. I was like, it's not worth it for a mark, but she was like, yeah, but... So I had to do it. I had to bloody do it. It is pain every minute of every day, bloody hell. The thing is too, like, I did really well in that. I had to, like, boost my own ego here, or, like, act like, you know, act like, I'm the best human ever or something. But, unironically, I think I did pretty well in my media exam, but the problem is, I literally just dropped 1 mark, and I checked Katie's work that she did. I think I checked Molly's maybe, or Miss, like, showed me Molly's work, and I would argue for my written bit, I did equally as good. So I have no idea why I was dropped a mark. In fact, no, Molly failed. Well, I think she has to redo it, because she dropped like 7 marks. She literally, she did the exact same work as me, like, same quality, and she failed. Which is scummy. Least scummy examiners. Even it's a epic fail. It is. It's a big fail. But anyways, let's crack on with this. Do any of you guys have any, like, suggestions? I'll do the heart, I guess. Because I did some research, okay, guys? Did some research, and I found that apparently the blood vessels in the heart haven't appeared much on these papers. Like last year, and I think the year before. I also heard that plant structure has appeared quite a bit. So actually, that means that you guys might not have to worry so much about the xylem and the phloem, but you may need to, you know, understand the differences between the veins, the arteries, the capillaries, maybe the digestive system. Definitely the heart though. And remember, I always draw the heart as a box because I can't draw a heart. You obviously have them 2 things going out, going in. So you have the vena cava there, the aorta there, the pulmonary vein, the pulmonary artery. And then you have your atrium and your ventricle. That right there, this bit here, your right atrium is where pacemaker cells are. If you get a question in an exam about that. How does the process of aerobic respiration differ between a unicellular organism? What the fuck? What is that? How am I meant to know that? I don't really get a microscope and say, hello, Mr. Cell, how many quantities of energy are you producing per millisecond? Thank God. I mean, yeah. I honestly, plant structure I like, because I've just learned it myself. Food tests may appear because, again, Mr. Hooper even said he hopes like the food tests appear, which means you guys better have remembered your bloody Benedict solution and you know, other stuff. You are Mr. Know-it-all. David asking the real questions, bro. I don't know, man. If that is an actual exam question, I would not be surprised because AQA is horrible sometimes. I was looking through the Wikipedia page of the exam boards. Turns out AQA, the only controversy they've ever had is 2022, where they gave out advanced information. But it turns out EdXL, this other exam company that you guys maybe do for history or something, that company is scummy. They've had a lot of controversies. They even stole some questions from like AQA at one point. So yeah, huge exam board drama. But yeah, pacemaker cells You could also get actual pacemakers, which are typically used for an irregular heart pattern. So if you talk about that, I guess. Then obviously you have stents and statins. Stents are like the metallic like tubes that go in your, I think, arteries to like widen them. It's obviously, the pros and cons are that the stents are typically last for quite some time. They have a chance of infection or your body like rejecting it, but it does widen your arteries for quite a while, a longer lasting time, but it requires a surgery, so it's likely more expensive. Statins are like the pills that you take, I think for cholesterol levels, I can't pronounce it, but basically they're obviously not a permanent solution. They're likely more cheaper, they're not invasive, unlike stents, which are invasive. And I think there's obviously a no chance of infection or rejection, but there could be other issues with like allergies, and it could be time consuming. You could accidentally miss taking one, which could be quite severe. So I think there's been 2 exam papers now where they've talked about the differences between stents and statins. So remember those. Obviously, one's invasive, one's not, and then you can just kind of branch out from there. How does a length of, what the hell does that mean? What is it? What is that, bro? Which exam board is controversial? EdXL, the Pearson one. I might have a book from them actually. Let me go have a look. Maybe? Oh yeah, I think here. Yeah, this one, Pearson. Because they do like history and stuff. They are not. What do you mean the 1st of March? That's not when the GCSEs come out, you batty crease. You're trying to deceive me, bro. Just for the record, I have not higher paper. What are you on about, bro? The higher paper and the foundation are released at the exact same time. I've had a process of bro, what does that mean? I guess actually a good topic to talk about would be what do you call it? That whole thing about, where is it? This damn question on, oh man, where is it? The whole topic on DNA and stuff, I hate it. I personally despise it. But unfortunately, I have to know it. But I don't know much of it, to be honest. I know some stuff. Like, I know that, like, it's the sugar and phosphate. As for bases, and they make up DNA. RNA is the code for, like, it's weird. All I know is that ribosomes synthesize proteins. That's it, really. But anyways, let's go do some actual stuff. I might turn off chat in a second, just to, well, turn it off temporarily, because I have no idea if you guys can see what I'm writing. But anyways, is that it for biology? Do you guys want me to go through the food tests or the enzymes? Or are you all happy with that? Because I'm relatively happy with it. I mean, in terms of enzymes, if you're doing like the lock and key method, you could talk about how the enzyme's active site binds with the complementary substrate. It breaks it down into useful products, and the enzyme becomes useful again. However, if it's not in optimal conditions, the enzyme becomes denatured, which could cause it to no longer bind correctly with the right substrate. But I'm sure all of you guys know that anyways. I I'm sure all of you guys know exactly what I'm on about. I'd hope. Anyways. So, let's do... Because again, I already have done food tests. Do I really need to go through... In fact, I'll go through dead quickly the enzymes, just in case any of you guys don't actually know it. Proteins, carbohydrates, and lipase. Honestly, the only bit that brain teases me is where they're found. That's the only bit that annoys me, because sometimes I'll forget one of the obvious ones. So, amylase, what does that do, guys? What does amylase do? Well, it's obviously an enzyme. What does it bind with? Well, it binds with starch, and it turns it into glucose. Okay? And a good way of remembering that is, basically, if you put like bread in your mouth, it kind of turns a bit sugary because glucose is sugar. You might get a blended question on how to test for glucose. And in that case, you should obviously refer to the Benedict solution. But you guys already know these stuff. So anyways, amylase, it turns starch into glucose. It is found in your salivary glands. Because remember, if you put bread in your mouth, It is also found in your small intestine, I believe. And your pancreas. I just remember anything with glucose or sugar has to be in the pancreas. Protease turns proteins to amino acids. Carbohydrates turns carbohydrates into simple sugars. And lipase turns basically fats or lipids into fatty acids and glycerol. Okay? So the proteins, I believe, are produced in the small intestine, pancreas, and the stomach, I want to say. The lipase, I believe, is only produced in the small intestine. It might be the pancreas. I might be mistaken. Carbohydrates, I know, is the small intestine, the pancreas, and the saliva glands. So, let me go check lipase quickly, because remember, even I can forget sometimes. And remember, this is quite a big topic. I'm just summarizing it briefly. Oh, and we should go through blood vessels quickly. Let me go quickly check, and then I'll go see. Actually, let me go see. I should have crawled in, gone, oh guys, I'm dead. What do you mean gunshot? Someone knocking on the door? It was not someone knocking on the door. So anyways, let me go check. Let me go check very quickly. I literally have COVID. This is the right book, right? I'm looking at my bookshelf. I honestly cannot tell. He's like Jeffy Dahmer. Who? If I love toxins, would you bro, where is this? Bro, where is the section on enzymes? Here. Okay, pancreas and small intestine. There we go. There you go, guys. Remember, obviously, this is important stuff here. Anything about the enzymes, food tests, I would say are worth your time. So we've already gone through the heart, so you guys should be able to answer these questions then. Okay. That's going to be the artery in this example. This will be the vein. And this will be the capillary. Okay. You guys now need to be able to know the differences between these. Name 3 differences between the artery and the vein. If you guys do not know this, know it. Number 1 is the fact that the arteries have a narrower lumen. Basically, lumen is like the interior bit of the blood vessels. Then obviously, arteries have thicker, muscular and elastic tissue because they need to be able to withstand higher pressures, higher blood flow. Veins have valves to prevent backflow. And that's basically it, right? That's literally every difference. I guess you could also refer to the fact that arteries go in to the heart, veins go out. Sorry, other way around. Arteries go out, veins go in. But if you didn't know that, you could say that, but I don't think you get any marks. The capillary is 1 cell thick. This allows it to easily diffuse ions in and out of it, so like into other cells, but also that's kind of it really. So cells can diffuse in or out, mainly ions. The capillaries are 1 cell thick. They are typically found in the villi or the alveoli, that's a word. Typically found in the alveoli or the villi because those features, those organs, need a constant supply of blood for a strong diffusion gradient. So there you go. That is that. Mr. Hooper said that could likely appear in the exam. And I know obviously people make memes about Mr. Hooper and stuff, but unironically, I think he does know what he's talking about. I feel like he's just kind of gotten screwed over. I mean, imagine having to teach the top set class, knowing that their previous teacher was Miss Collinson. Like, obviously, he's not going to stand up very well against the test of that. But yeah, that's kind of it. So anyways, we've done biology. I think that's it. I think that's everything that I wanted to talk about. Let me just go through my revision guide and see. Oh, cancer, I guess we could quickly talk about. There's the 2 types, the benign, where it only grows until there's no more room, Typically, you only like a bit of tissue, and then it will stop. And it's not cancerous, it doesn't spread. But then malignant is where it grows and spreads to neighboring tissues. It can break off and invade other parts of the body by traveling in the bloodstream. And of course, they are dangerous and can be fatal. Which is self-explanatory. I remember a diagram in one of the mock exams that we did showed a lung with a couple of spots on it, and it said these spots are cancer. How do you know that the tumour is malignant? And you had to talk about how actually it's because it grows in multiple spots. So if you didn't know that, then be sure to write that down. Right. There we go, benign. Okay. So let's go do some chemistry for 15 minutes. Obviously, the topic that everyone hates, and yet I kind of like, is electrolysis, where you obviously have your positive anode, your negative cathode, oil rig, the fact that Luke Kelly and breakdown, ionic breakdown. Honestly, at this point, I'm not even trying to explain it, because if you guys don't know this by now, check the Google document, like I've said a trillion times. You're only missing out if you're not part of it. I'm just jotting down some ideas here. Obviously, bauxite, molten cryolite. You guys get the deal. In terms of organic chemistry, you obviously have your alkene, alkene, alcohol, and carboxylic acid. Obviously, alkanes, group, some functional group, sorry. Alkane's functional group is nothing. Remember, carbon can bond to something 4 times. Alkene's functional group is that it has a double carbon bond, which means these are only going to have 2 hydrogen bonds each. Alcohol's functional group is that it has an OH. And the carboxylic acid functional group is that it has an OH double bond, an H bond, an O bond, because it's COOH. And remember, because it's an acid, it disassociates to give you H+. But you guys knew that already, I'm assuming. What you probably didn't know is how to get from X to Y. How do you get from an alkene to an alcohol, and then an alcohol to a carboxylic acid? Riddle me that, guys. How do we do it? Well, obviously, to get from an alkene to an alcohol, what are you adding? Well, you're breaking that double C bond, okay, and you're adding 1 hydrogen there, and you're adding 1 oxygen and 1 hydrogen. So you're adding 2 hydrogen, 1 oxygen. Hang on a minute. H2O. You're adding water. More kind of specifically, you're adding steam, hot water, scalding water. So you add the scalding water, and you typically also use a catalyst. Remember, a catalyst increases the rate of reactions, and that is phosphoric acid. Okay. Finally, to get from an alcohol to a carboxylic acid, you need an oxidizing agent, because all you're doing is you're just adding a double bond of oxygen. You're adding an oxygen there. So you use an oxidizing agent. I can't really name too many oxidizing agents off the top of my head. If you guys know any, that'll be good. But that's that. I mean, obviously there's the whole thing of esters, which is like an alcohol... Oh, addition polymerization, which only creates 1 product. Condensation polymerization, which creates 2. It often involves, condensation. Can't remember the other thing off the top of my head for that, but that is an interesting thing to note. It's typically water is that second smaller molecule. Oh, let's do something super basic. Endothermic is the name. Name the differences. Well, obviously, this one starts out high and it goes to low. This one starts off low and it goes to high. The activation energy is the amount of energy required of a reaction, which I'm assuming in that case would be that. The overall energy change would be that. That could give you a lot of marks in an exam. Just remember, when talking about endothermic, energy is taken in from the surroundings, energy is released in exothermic. So for example, photosynthesis is an example of endothermic, I believe, because you're taking in energy from the surroundings, light energy, and then obviously you're creating stuff with it. Okay, here's another quick biology thing, actually. Guys, seriously, guys, and anyone could answer this, okay? If I got a light, okay, there's algae in a bottle. If I got a light here, and the light was warm, alright, if the light was warm, what do you think would happen to the tube? Like, what? So there's algae in a bottle of water. What do you think would happen? Well, obviously, temperature is a limiting factor of photosynthesis, which means more oxygen is going to be produced because temperature is increased. And I did not know that as of a week ago. I forgot that if you increase the temperature of something, more oxygen bubbles are produced because it's a limiting factor. How would you stop that, guys? If I don't want this light to produce heat, how would I stop that? Literally, the easiest answer, use an LED. And that could be 4 marks in an exam there that you didn't know before. Goodbye. Thanks for watching. Check the Google document if you haven't already. Seriously, guys, it is a godsend. I have learnt so much just from my own independent revision, and I'm helping you guys by spreading this information. On a completely serious note, guys, I do understand that it is getting very close to the exams now. I myself am worried sick about some of the questions that we're going to get, because I swear down there's going to be some vile ones. Like, I don't know, man. I feel like biology paper 2 is going to be repulsive as hell. But I'm doing these streams to try and help explain what I know. But also, this is like a collaborative thing. If you guys know something that I don't, let me know. If I know something that you don't know, write it down. Go on the Google document and whatever. Anyways, I'm going to go now because I got some stuff to do. Goodbye.